Hello everyone, welcome back to the class. In our previous class, we learned about role of RB protein in cell cycle regulation. So we started our discussion by defining what is RB protein, and RB protein has been coded by a gene, that is RB1, retinoblastoma 1. It is a tumor suppressor gene, which genes whose inactivation contribute to the development of a tumor. It codes protein called PRB, which is a nuclear protein of 110 kilodaltons, and the protein encoded by RB1, that is PRB, is a negative regulator of cell cycle. Loss of function mutation of RB1 will result into retinoblastoma and many other tumors. So RB1 is responsible for suppressing the tumors. So inactivation, if there is absence of function of RB1 gene, it leads to retinoblastoma and may cause tumors. So what is the function of RB protein in cell cycle regulation is majorly or phosphorylation. Active state of PRB protein, unphosphorylated state of PRB protein, hypophosphorylated PRB proteins will bind to the transcription factor E2F, which represses the gene transcription which is required for transition from G1 to S phase, and it halts at the G1 phase itself. E2F acts as a transcription factor and induces many genes to transcribe. Once, if there is no binding with PRB, E2F transcribes many genes. The product of these genes are required for S phase in the cycle. As E2F is busy bound to PRB protein, it is unable to activate the genes of S phase of cycle. When PRB protein binds with E2F, E2F activity is suppressed. The phosphorylation of PRB protein reduces the affinity for E2F. PRB protein is a substrate of cyclin-CDK complex. The PRB protein is phosphorylated by cyclin-CDK complex, especially cyclin-D-CDK 4/6 and cyclin-E-CDK2 in G1 phase. It allows it to E2F after phosphorylation of PRB protein by cyclin-D-CDK 4/6 and cyclin-E-CDK2. It allows E2F to heterodimerize with the factor DP and initiate the transcription of genes which are whose product are necessary for S phase cycle. It induces expression of critical late G1 genes. So the product of the G1 genes are required to prepare the cells for entrance into S phase. So this is the image which depicts here PRB protein and E2F has formed a strong connection in the mid G1 phase. When cyclin-D-CDK 4/6 is phosphorylating RB protein, so after phosphorylation it leaves E2F behind, and E2F is the affinity of E2F towards PRB protein is reduced. E2F will bind to DP and it forms a heterodimer. E2F DP complex will induce expression of critical late genes which are the products required for S phase of the cycle. Or hypophosphorylated PRB protein, which is active, it blocks cell cycle progression. Cyclin D and E control the degree to which PRB is phosphorylated. Cyclin D-CDK4 initiates the hypophosphorylation in the mid G1 phase. It is a prerequisite to the restriction point. The R point determines whether the cell cycle should initiate, continue, or pause. Cyclin E level increases dramatically. The role of cyclin E and the CDK2 drives PRB phosphorylation to completion, leaving PRB in its hyperphosphorylated state. R point, this point is also known as restriction point. It is a point which represents the cell which commits itself to complete the remainder of the cell cycle, or alternatively to remain in G1 and possibly retreat from the active cell cycle into G0 quiescence stage. There are two other members of RB protein which has been recognized: pocket protein family. Pocket is because it is having a conserved domain called pocket, which interacts with LXCXE motif found in some viral proteins. The two other members other than RB1 family are RBL1, RBL2. RBL1 gene codes for protein P107, RBL2 codes for protein P130. So today we will be discussing about role of another protein, that is p53 protein, in cell cycle regulation. So to introduce p53 protein, so to give an introduction, p53 is another tumor suppressor gene. It is like the protein we discussed earlier, that is RB protein. So the tumor suppressor gene TP53, tumor p53 gene codes for p53 protein, which is the gene TP53 is the gene which codes for p53 protein. The apparent molecular mass of this protein is 53 kilodalton. Okay, TP53 gene is more or less like RB1 gene, as it is a tumor suppressor gene. Its activity, when it is in active state, it stops formation of tumor. Therefore it is known as tumor suppressor gene. It stops formation of tumors. In humans, this TP53 is present in the chromosome 17p13.1 and encodes a polypeptide chain of 393 amino acid residue. So it encodes a polypeptide chain of how many amino acid residues? 393 amino residues. So p53 is a protein which is coded by TP53 gene, which is like RB1 gene, as it is a tumor suppressor gene. Its activity stops the formation of tumors. It is located in human chromosome 17p13.1 and encodes a polypeptide chain of 393 amino acid residue. p53 protein acts as a transcription factor. So it is a transcription factor which regulates expression of genes, which are involved in DNA repair, cell growth, apoptosis, antioxidant defense, and senescence. It regulates, this p53 protein regulates expression of several genes involved in DNA repair, cell growth, apoptosis, antioxidant defense, and senescence. Because because of mutations in TP53 gene are found in most tumor types and are involved in the complex network of cellular events. So this TP53, which is a gene, is known to be a guardian of genome, and also it is known as cellular gatekeeper. In normal cell, this p53, whose function is to stop the formation of tumor. If it is inactive, what happens? This is inactivated by its negative regulator, that is MDM2. MDM2 makes p53 inactive. Full form of MDM is mouse double minute. P53 is inactivated by negative regulator. MDM is a regulator which inactivates the p53. MDM2 functions as, this MDM2 functions as E3 ubiquitin ligase. This causes proteasomal degradation of what? Proteasomal degradation of p53. So p53 is inactivated by negative regulator MDM2. The MDM2 protein acts as a E3 ubiquitin ligase, and it causes proteasomal degradation of p53. Consider there is a DNA damage. Or other stress or various paths leads to dissociation. So by convention, p53 is inactivated by MDM complex. So when there is DNA damage, the p53 and MDM2 complex breaks down. So it leads to dissociation of this complex. This is the first step when DNA damage occurs. So after this p53 dissociated with the MDM2, it becomes free p53. It is now free p53. This will induce cell cycle arrest, and it allows either repair and survival or apoptosis or death. Okay, so this is the thing. For example, DNA damage activates p53. So consider there is a DNA damage. What I said now, DNA damage will activate p53. What if there is no damage? Undamaged cell. So the p53 is highly unstable and present in low concentration. This is largely because it interacts with MDM2. So there is no free p53 present. So p53 interacts with MDM and which causes ubiquitination of p53. As I mentioned, MDM2 MDM2 is a E3 ubiquitin ligase, so it ubiquitilates this p53, and it destructs this p53, proteasomal destruction. Consider p53 is phosphorylated after DNA damage. This phosphorylation reduces binding with MDM2. As it is associated with phosphate group here, it will not bind to MDM2. So therefore we can say that phosphorylated p53 will be active to perform its function. So once it binds with MDM2, once p53 is bound to MDM2, it helps in degradation, we know. So what happens in this case when phosphorylation of p53 takes place? It decreases the rate of degradation. It decreases the degradation, and by decreasing degradation of p53, by default we can say there is increase in p53 concentration in the cell. So there are more number of p53 as it has escaped from the degradation with MDM2, as it is bound to phosphate group. Now the cell is having more number of p53. Consider now there is a double strand break in the DNA. So it is a type of damage, right? So protein kinase ATM is activated. First ATM is activated. This activated ATM in turn activates CHK2 kinase. Where did you learn this pathway, or what is this pathway known as? ATM pathway. That is ataxia telangiectasia mutated, which will recognize the double stranded break. After the activation of CHK2 kinase, so now CHK2 kinase is activated. Both ATM and CHK2 kinase phosphorylates p53. It phosphorylates p53 at a distinct site. Okay, the phosphorylation of p53 on amino acid residue. So if this is a p53 and p53 protein is having a N-terminal domain, and in N-terminal domain there is a particular amino acid to which the phosphate group is added. So the phosphorylation happens at the amino acid residue on the N-terminal domain by kinase such as ATM and CHK2. Now p53 is phosphorylated. Okay, at this stage the p53 will not bond with MDM2. It prevents the association of MDM2 with p53. At the same time, the ATM kinase, which was responsible to activate the CHK2, which in turn phosphorylated this p53. So this has the capacity phosphorylate even MDM2. Even MDM2. Okay. So what is the purpose of phosphorylating MDM2? So it inactivates, so it makes functionally inactive. It makes functionally inactive. It makes MDM functionally inactive. Once MDM is phosphorylated, it will also not associate with p53 and to help in proteasomal destruction of p53. So ATM can phosphorylate p53 also, and ATM can phosphorylate MDM2 in a way that causes its functional inactivation. As a consequence of phosphorylation of both p53 and MDM2 fails to initiate. So fails to initiate ubiquitin degradation of what? Ubiquitin degradation of p53. This also helps in increase in p53. As p53 escapes destruction, the p53's concentration will be more in the cell. Once present in substantial amount, the p53 is poised to evoke a series of downstream response. This p53 protein stimulates transcription of genes encoding CKI protein. So I'll write in this way. So p53 helps in transcription of genes such as p21, which is a gene encoding CKI protein. So we can say p53 allows the transcription of gene encoding a CKI protein, that is p21. So now p53 has helped in transcription of gene which codes for CKI protein. That protein is p21. So what is the function of this p21, which is a protein, CKI protein? It is a CKI protein. So this protein binds to G1-SCDK and SCDK complex. Okay, it binds to G1-SCDK and SCDK complex, and this binding inhibits activities of what? G1-SCDK and SCDK. So what happens if there is inactivation of G1-SCDK and SCDK? So it helps to block entry into cell cycle. As there is a DNA damage, the cell should not proceed to next process, which is regulated by SCDK and G1-SCDK, which favors that these complex like G1-SCDK and SCDK will help the continuation of cell cycle. It is very much needed at this stage. As there is a DNA damage, the cell should not continue the process. It should halt at the process wherever it is. It should halt. It should stop at the stage. So therefore this p53 regulates, or it helps in stopping, or it helps in blocking the entry into the cell cycle. So there is a DNA damage. And in response to DNA damage, p53 level is high in the cell, and this p53 encodes P21 encodes the gene which codes P53 stimulates the transcription of gene which encodes the CKI protein, that is P21. P21 will bind to G1S-CDK or S-CDK complex. Binding with this will cause inactivation of this helps in arrest of cell cycle. It arrests the cell at the G1 phase of the cell cycle. Hence if all the repair has been done in this DNA, the cell divides normally and completes the cell cycle. However, if the cell still contains mutated or duplicated DNA sequence, it dies by suicidal apoptotic mechanism to prevent its proliferation. Imagine if there is a mutation or if there is a loss of function of P53. Okay, what happens? The arrest of G1 will not occur, and the cells that have mutated genomes proliferate. So genomes proliferate and it become cancerous. Okay. So to diagrammatically understand what's happening here, what's the function of P53 in cell cycle regulation. So consider a double strand DNA and there is a break here, double stranded break. So this double stranded break has been identified by our ATM. Okay, ATM. So this ATM is activated and makes CHK2 to activate. Okay, and we know there is P53. I'll write it in a different color. So there is P53 which is associated with MDM by convention. P53 when associated with MDM2, will it form or will it be free? Will it be active? No, it is inactive. So when CHK2 is activated, this will allow MDM to dissociate from the P53, making P53 free. Then P53 acts as a transcription factor for P21. Transcription factor for P21 genes, and P21 genes will synthesize P21 protein. This P21 protein acts as CKI. Cyclin. What is the full form of CKI? CDK inhibitor proteins. It inhibits the function of CDKs, right? So this CKI will bind to G1S CDK and SCDK. What happens if it binds? It makes it inactive. And what happens if G1 or SCDK or SCDK is inactive? It gives time. It arrests the cell cycle at G1 phase and all the repairs, and it gives time to repair all the DNA damage. So numerous reports have been described that mechanism by which P53 induces apoptosis. So there is a process which is known as P53 mediated apoptosis, which involves the coordination of transcription independent functions of P53. P53 acts as a transcription factor for numerous genes which are helpful for synthesizing p21 protein, for example. So it is involved in apoptosis such as puma. So it is helpful in transcribing PUMA and NOXA, which are the genes involved in apoptosis. In addition to these nuclear activities of p53 is also having cytosolic activities that can induce the apoptosis in a transcription independent manner. And p53 can induce mitochondrial outer membrane permeabilization, which we call as MOMP. So this p53 induce mitochondrial outer membrane permeabilization, thus leading to release of cytochrome C from the mitochondria. So let us see one by one. Role of p53 protein in cell cycle regulation, which is the p53 protein is it is a product of TP53 gene. It is a tumor suppressor gene codes p53 protein, which stimulates transcription of gene encoding CKI protein, which is p21. CKI protein binds to G1 or SCDK and complexes and inhibits their activity, thereby helping to block the entry into the cell cycle. Hence, in response to DNA damage, p53 level increases, arrests the cell cycle at G1 phase of the cell cycle. If all repairs are made to DNA, cells divides normally and completes the cell cycle. If cells still contain mutated or duplicated DNA sequence, it dies by a suicidal apoptosis mechanism to prevent its proliferation. So here see the normal cells and cancerous cells. In normal cells, p53 is highly unstable and present in very low concentration, as it is inactivated by MDM2, which is known as mouse double minute 2. So mouse double minute 2 functions as a E3 ubiquitin ligase. See here, ubiquitin chain has been inserted to p53, making it inactive. So in cells which contains mutated p53 or lost p53 genes, arrest at G1 phase does not occur. Cells that have mutated genomes proliferate and becomes cancerous. DNA damage or stress. If there is a DNA damage or stress, various pathways dissociate p53 from MDM2, making p53 free. Okay, p53 is activated by phosphorylation by who? CHK2 kinase. CHK2 kinase is activated by who? ATM. So ATM, when there is a DNA damage, first ATM is activated, then it activates CHK2 kinase. CHK2 kinase phosphorylates p53 and makes it active. This active p53 will what it does will help in synthesizing p21 protein by acting as a transcription factor for p21 gene, and that binds with G1 or SCDK and SCDK and it makes G1 SCDK inactive. So this is the image how DNA damage arrests the cell cycle. So there is a DNA damage, ATM kinase activated, and it activates CHK2 kinase, and that p53 is phosphorylated by CHK2, allowing that to become stable and active. Active p53 binds to the regulatory region of p21 gene. Transcription allows the synthesis of p21 protein. After translation, the p21 acts as a CDK inhibitor protein, where the active CDK G1 SCDK and SCDK becomes inactive. ATM kinase phosphorylates. So ATM kinase can phosphorylate MDM2 also, and it causes functional inactivation. See here, MYC. Okay, And here you can see p53 degradation, as there is active MDM2, it makes inactive MDM by phosphorylating MDM2. p53 induces apoptosis. p53 mediated apoptosis involves the coordination of transcription dependent and transcription independent function of p53. p53 acts as transcription factor for numerous genes involved in apoptosis such as PUMA and NOXA. p53 also has a cytosolic activities that can induce apoptosis in a transcription independent manner. It can induce mitochondrial outer membrane permeabilization, that is MOMP, thus leading to release of cytochrome C from the mitochondria. So this was about role of p53 protein in cell cycle regulation. In our next class, we'll be learning about replicative senescence, and we'll start the mechanism of cell cycle, that is part 2 of the cell cycle. So thank you.